All right, can you see the screen?
Yes.
All right, good. Good morning, everyone. Welcome to our department seminar. Today's seminar is special. It's the 23rd Moraj Memorial Seminar. This seminar was created in 1998 to honor Dr. Karl Moraj. He had an extraordinary career. He's an eminent entomologist, virologist, and plant pathologist. So this award, this seminar was established to recognize distinguished alumni, postdocs, and faculty that's associated with Rutgers. However, over the years, we ran out of those candidates. We started to try those eminent scientists outside the university. So today we invited Dr. Chaiyan Lee to be our seminar speaker. I'll give you a brief background about Dr. Karl Moraj. So he was born in Vienna when his family fled from the outbreak of World War I. After about 3 years, when he was 3 years old, basically, then his family moved back to Poland, where he studied for agriculture in Warsaw University. However, the World War II outbreak that displaced him and his family. So he escaped to Sweden. Well, his parents' brother and more than 120 relatives, unfortunately, perished during the World War II. So he was a very lucky person, he always said. He immigrated into US in 1947. Then only after 2 years, he got a PhD from Columbia University. After that, he worked at 3 different institutions. Then he finally settled at our department in 1984. So he is, I would say, a very talented and multifaceted person. He's a gifted pianist. He enjoys photographing. He is basically a very talented person. And he also worked very hard. He, at 98 years old, he still drove from New York to New Jersey, come to work. And I remember that at 98 years old, he still traveled to Tanzania for a meeting. And for 30 hours trip, he was like on economy class. He always said that in order to live a long life, never stop working. So he's really an extraordinary person. So for more details about him, you may want to read this book, The Thorny Road to Success. So today I'd like to introduce our speaker, Dr. Chaiyan Lee. So Chaiyan got his bachelor and PhD degree in University Sains, Malaysia. Then after that, he continued to work at the university from the rank of lecturer to full professor. During that period of time, he received numerous awards, including the called the Young Scientist Award and Top Research Scientist Award from Malaysia. Then in 2019, he accepted the position of endowed presidential chair in urban entomology at the University of California, Riverside. His current research focuses on the biology, behavior, and management of urban pests. He had a very broad interest in all urban pests, which is similar to my current research area. So I'm really excited for him to give us the memorial seminar today. So now I'm going to stop sharing, and Chaiyan, you can go ahead.
Okay. Thank you so much, Chang Lu, for the kind introduction. I'd like to share the screen now. So first of all, I would like to thank Chang-Lu Wang and also Rutgers Entomology Department for the invitation to deliver this Mara Moraj Memorial Lecture. So today I'm going to speak on the global perspective of insecticide resistance in bedbugs. I have a total of about 60 slides, which I'm going to try to cover within 50 minutes. So this is a brief outline of my presentation. I will first talk about bedbug infestation around the world, the control strategies, insecticide resistance issue, resistant management strategies, and what lies ahead for the future. So if we look at bedbugs, this has become one of the most important urban insect pests in many parts of the world. If we estimate in terms of fiscal impact, about $1 billion is being spent each year in the US for bedbug management. Now, the infestation of bedbugs is no longer a problem in residential and public accommodation, but it has since become an issue also in public transportations, even in laundry and many others. So over the last 10 years to 15 years, we have seen a drastic increase in bedbug market segment, especially in the pest management industry around the world. So bedbugs would be able to spread very easily from places to places, especially through hitchhike. And this particular video, it's actually taken in a hotel in Malaysia, where there had been serious tropical bedbug infestation. Now, if you track the peer-reviewed publication of bedbugs through Web of Science, you can see an exponential increase since 1912. And of course, over the last 10 years, there has been an explosion of publication of peer-reviewed papers on bedbugs. So this would be the telltale sign of the person who get bitten by bedbugs, but not everyone would be showing a rash or a symptom like this, because this only happen in people who are hypersensitive, but to some other people, such reaction may not happen. Globally, there are two major species of bedbugs that are causing the infestation. The first one is the common bedbugs, that is Cymex lectularius, and the second is the tropical bedbugs. So in terms of differences, morphologically, they are very similar to one another. If you have not seen a tropical bedbug before, it is hard to tell a common bedbug from the other. Or if you are not seen a common bedbug before, it is hard to tell a tropical bedbug from the common bedbug. But if you put both side by side, okay, if you look at the pronotum, the pronotum or the prothorax of the common bedbug has a larger lateral flange compared with the tropical bedbug. In terms of global distribution, this is a map that I update on February 22nd, 2021. So you can find that tropical bedbugs generally hover around the tropical region, but more recently, they have also been found in different parts of Europe. Okay, so it is a problem, especially in Southeast Asia, in some places such as Brisbane or Queensland, Australia, they are there. In the southern Japan, they had been found. And of course, in the West Africa, both species are also symmetrically coexist. Now, but more recently, the tropical bedbug also has been found in Europe. Okay, but if you look at the common bedbug infestation, they pretty much found around the world. Okay, with exception to the tropical part of Asia. So these are two papers that were recently reported on tropical bedbugs in Russia. And what is interesting with this is in some parts of Russia or where the infestation are being found, they could only find Cimex hemipterus, that is the tropical bedbug, but there are no Cimex lactularius, the common bedbug, despite in the past the common bedbug has been found in those locations. Tropical bedbugs have also been reported in Italy more recently. So in the US, the common bedbug is found throughout the continental US as well as also Hawaii, but tropical bedbugs are only found in Hawaii and Florida. But having said that, I believe that if we were to examine the sample more carefully, I'm sure that tropical bedbugs would be found in other southern states as well. Now, in terms of biology, between the common and tropical bedbugs, both undergo nympha stages. There are 5 nympha stages. At a certain period, at a certain temperature, that is between 27 to 30 degree C, you would find that the life cycle is relatively similar. In terms of lifespan, at 27 degrees C, you would find that in terms of lifespan, they also resemble one another. However, tropical bedbugs have been found to be able to climb better on smooth surfaces compared with the common bedbug, so making them able to escape easily from most of the pitfall traps that are used for monitoring. Okay, as you can see right here, there are 4 different brands of pitfall traps being used. So the tropical bedbugs could escape. For example, for this verified that is no longer in the market, they could escape up to 80% when contained in the pitfall traps within a short period of time. But for common bedbugs, you would find that most of them could be contained in the pitfall traps. So, and the reason to that is when we examine the tenon hairs on the tropical bedbugs, there are significantly more tenon hairs on the legs of tropical bedbugs compared with the common bedbugs. Both species are able to develop resistance to insecticide, especially towards pyrethroids, organophosphate, carbamates, and neonics. And this wood clouds is actually constructed based on peer-reviewed papers that were published over the last 15 years. As you can see, most of the insecticide reported here are actually pyrethroids. So what actually caused the global invasion of these bedbugs, or what we call modern bedbugs? Now, resistance to pyrethroid are documented for both species. And interestingly, both species have also been shown to have different forms of KDR as well as metabolic resistance, although the specific of these resistance mechanisms are different. So the question is, why would both species of bedbug that has actually diminished in importance back in the 60s become prominent again in the 90s, with population of both species popping out in different parts of the world, showing widespread pyrethroid resistance? So could all these modern pyrethroid resistance population originated from locations where both species existed sympatrically? And could that be Africa, where the ancestor of all these modern bedbugs that live sympatrically were initially subjected to selection pressure, to DDT, and then later to pyrethroid due to indoor residue sprays, and also pyrethroid impregnated bed nets that is used for malaria control. So this basically put out a out of Africa hypothesis, but until today, we have not been able to prove this. So let us talk about insecticide resistance. So Resistance is an inherited ability of a few population to withstand an insecticide concentration that will normally kill a normal population. So in the laboratory, we normally would define this as an increase in insecticide concentration, dose or time required to achieve mortality of a few collected population in comparison to a laboratory strain. Okay, but in the field, this would be defined as a repeated failure to control a population using a labor rate of a product. So what actually drives resistant development? So there are a number of factors. First, of course, is repeated use of the insecticide that are closely related. Second, of course, it's when a large population, when a large percentage of the population are being treated. So when a large percentage of the population being treated, they exert selection pressure to that population and select out the resistant individual. And of course, it is also due to low human tolerance threshold. So because we have low tolerance threshold to blood-sucking insects, so the more likely we would want to put out any control program or any control measure to manage them, and hence the pest population would be able to develop resistance at a faster rate. Of course, through thorough application methods that leave no untreated hiding pests, it's also one of the factors. And lastly, the continuous selection of insecticide for many, many generations. But having said that, not all insecticide treatment failures could be due to resistance. There have been many instances where incorrect dilution, improper application techniques, poor trained staff, and of course, when harborage are not being treated, has been found that is causing the treatment failures. So hypothetically, when a populations of bedbug, an original population of bedbug or a wild population of bedbugs, they are bound to have some individuals within that population that are having the resistant gene. So as we continue to select that population with the same insecticide or insecticide from the same group over and over for many generations, there will be a point when the population become predominantly consists of individuals that are carrying the resistant gene. And that will be the point when we will see control failure. So worldwide, we believe that this modern resurgence of both species of bedbug, it's due in very large part to insecticide resistance. So there are two major reviews on insecticide resistance in bedbugs that have been published over the last 5 years. And for those who are interested, you can refer to this paper by Dang in Parasite and Vector that was published in 2017. And also in this book, Advances in Biology and Management of Bedbug, there is a chapter by Alvaro Romero on insecticide resistance. So if we were to look at insecticide classes used against bedbug around the world, you would find that predominantly most of the synthetic organic insecticide have problems with resistance, okay? Ranging from chlorinated hydrocarbon all the way until neonicotinoids and even pyrrols. So cases of resistance has been reported on all these different classes. So far, IGR are still okay, that there have not been any reports on insecticide resistance, botanicals, as well as also inorganic insecticide. So in terms of resistant mechanisms, we can divide them into 4 major mechanisms that has been found in bedbugs. So the first one is penetration resistance. And this, of course, because to kill a bedbugs, the insecticide would need to penetrate through the bedbug cuticle to get into inside the body of the insects. But the cuticle is a waxy barrier. So if the cuticle is thickened, then this would reduce insecticide penetration. So generally, penetration resistance could confer resistance across a wide range of insecticide classes. And one of this interesting study was conducted by Dr. Stephen Dodgett's group in Australia. So what they have done is they have subjected the bedbugs to pyrethroids, and those that got killed are considered the intolerant one within 2 hours, and then those that are killed between 4 and 24 hours are the tolerant ones, and lastly, the resistant ones are those that survive more than 24 hours. What happened is he then took all these that are dead within 2 hours, compare with the one that are dead in 4 and 24 hours, and subsequently also the one that are dead after exposure of more than 24 hours, and compare in terms of cuticle thickness. And what they have seen here is they have significant differences in terms of cuticle thickness. So using the same strain of bedbugs, they have also found that these strains at sublethal concentration, the pyrethroid resistant strain, they will be able to survive even to inorganic dusts such as silica gel and diatomaceous earth. The second mechanism is metabolic resistance, and this is due to increase in enzymatic activity that will lead to increased detoxification of insecticide due to the overexpression of the gene. So we have 3 groups of enzymes. The first one, cytochrome P450, esterases, as well as also GST. So if you look at the detoxification enzyme and the insecticide classes that they could confer resistance to, you would find that one of the major issue is actually the monooxygenase or the cytochrome P450. So practically, it could confer resistance to most of the synthetic organic insecticide that are used on bedbugs. Esterases only confer resistance towards OP, carbamates, and pyrethroids, and GST are mainly on OPs and pyrethroids. Next, we have target site insensitivity, and this would be when the sites where the insecticide attack become modified, and insecticide will have less binding affinity towards this site, and hence they are less sensitive. And the insects, when they get exposed to the insecticide, they will not get killed. So far, in the urban pest world, we have detected 4 types of target site insensitivity, but in bedbugs, there are only 2 that has been reported so far. There is KDR resistance, as well as also altered acetylcholine esterase. Okay, RDL mutation has not been reported, and also the altered ACHR has not been reported so far in bedbugs. So you can see that for KDR resistance, this would confer resistance towards the chlorinated hydrocarbon group, and also, but of course, this is only limited to DDT. Deldrin would be conferred, the resistance to deldrin would be due to RDL, okay? But in the case of bedbugs, only to DDT. The KDR will confer resistance to, and of course also to pyrethroids. So the number of KDR mutations that has been reported in Cymex, there are many, but I'm just going to narrow it down on important ones that are being highlighted here. For Cymex lactularius, it will be the V419L, and also the L925I. Okay, these are the 2 most common ones that confer resistance to the common bedbugs. On the other hand, for tropical bedbugs, it will be the M918I, as well as also the L1014F. These are the 2 most common ones. And sometimes they will exist singly, sometimes they will exist in pairs. So in the situation when both coexist together, normally the level of resistance are much higher. On the case of behavioral resistance, little is known on these mechanisms, but there have been reports showing that resistant bedbugs may detect the presence of insecticide and then avoid coming into contact with the treated surface, or being repelled away from coming into close to the treatment. And this, it's still relatively or very limitedly known, and more work should be done in future on behavioral resistance in bedbugs. So let me show you some of the recent published report on bedbug resistance around the world over the last 3 years. So if you look at all these published reports between these common bedbugs as well as also the tropical bedbugs, most of these reports are reporting resistance to pyrethroids. Now, despite widespread pyrethroid resistance, unfortunately, pyrethroid continue to be used against bedbugs. And this is irony. Okay, so this shows the report on Cymex lactularius. So you can see in Europe, they tested 105 population from 14 countries, and about 94% of all those population have KDR mutation, okay, at 925 sites. In the US, for example, they collected from 97 cities across 7 states, and still they found high prevalence of KDR mutation. Okay, and similarly in France, they are also finding many of these collected population with KDR mutations. So in the tropicals, you would find that very similar situation, with regards to pyrethroid resistance. So most of the population collected are also showing KDR mutations, as well as also metabolic resistance due to cytochrome P450. So, of course, when we have resistance, we will have to talk about resistant management strategy. Now, the whole idea about resistant management is to prevent and delay resistant development. So in practice, what we want is we want to prevent the selection pressure from a single insecticide or a single insecticide class or that particular mode of action. Now, to achieve these, we will have to prevent any single mode of action use. And of course, we would try to decrease the frequency of single mode of action use. Now, if we look at the chemical control methods against bedbugs, okay, in terms of formulation, these are the different types of formulation that are widely used against bedbug control. Or on bedbug control, you can see liquid spray, pressurized aerosol, dust, fumigants. Then we have the insecticide impregnated fabric, repellent, and also a total release fogger, or sometimes they call it the bug bomb. Okay, so in terms of ability to manage or to kill the bed bugs, you will find that actually for liquid spray, which is widely used, they really have very low effectiveness. I'm going to show you data on insecticide impregnated fabric, as well as total release fogger. But all these three different formulation or three methods of chemical control, essentially they are less effective or have low effectiveness. This is one of the unpublished data of my doctorate students in University of Science Malaysia. And in this study, she evaluated one of the popular permethrin impregnated mattress liner that is being used, especially in public accommodation, hotels, motels, to prevent bed bugs. As you can see, with the susceptible strain, we can achieve 100% mortality in 100% mortality, and the LT50 for this is 19 minutes. So imagine that the bed bugs are actually continuously exposed for 96 hours. LT50, 19 minutes, okay? But on the other hand, okay, with the resistant strain of the tropical bed bugs, okay, as you can see right here, these are the different resistant strain. Some of them only had less than 5% mortality, even being exposed for 96 hours, okay? And of course, in terms of LT50, it reached a stage where because they are so low number, we are unable to generate the LT50 value, okay? But this is very common, and still, despite the ineffectiveness of product like this, they continue to be used in the market. So one of the method of resistant management is the use of insecticide rotation. So this approach basically rotates insecticide of different mode of action in sequence once every quarter. And it is important to know that many of the different brands actually contain the same insecticide, different brand names. And also different chemical names may be of the same insecticide class and having the same mode of action. But unfortunately, for bedbug management, there have been very limited number of options available for rotation. And based on what I have shown earlier, okay, because most of the classes of insecticide that are used for bedbug control, there have been reported cases of resistance. So we really have very limited option when it comes to insecticide rotation. So one of such option would be to have a pyrols used for 3 months, followed by botanicals or desiccant, and then followed by a mixture of pyrethroids and neonics for 3 months, and then the cycle continue. But as I said, really do not have much option. So insecticide mixture, it's a common scenario these days. There have been a number of companies that will have come out with mixture product. For example, by Bayer, we have beta-cyfutrin. So it's a pyrethroid with a neonicotinoids mixture. We also have Syngenta coming out with Tandem, also a pyrethroid and a neonicotinoid mixture. We had Transport by FMC, that is also a pyrethroid and neonicotinoid mixture. And lastly, by MGK, Crossfire, there is a pyrethroid, a neonicotinoid, as well as piperonyl butoxide. So what is interesting is most of these products have a pyrethroid and neonicotinoid combination, okay? But unfortunately, we have also seen that bedbug has become resistant to the mixture. So in this particular study, you can see that the susceptible strain would achieve 100% mortality, but for the resistant strain, we are unable to achieve even 50% mortality right here, okay? But if we were to have piperonyl butoxide added to this mixture, you would find that mortality could be increased to nearly 90% or even 100%. So this is because cytochrome P450, as I mentioned earlier, this is one of the major metabolic resistance mechanism that confer resistance across many of these different insecticide classes. So if you use a piperonyl butoxide that is known as a synergist or a cytochrome P450 inhibitor, we would be able to achieve better control of the bedbugs when used in combination with the mixture. Other options, of course, there have been biopesticides now in the market, microbial insecticide that contain entomopathogenic fungi, Beauveria bassiana. And this is one formulation. We call it the ready-to-use RTU formulation, where it contains spore and also oil mixture. But unlike insecticide, the microbial insecticide normally would take time to kill the bedbugs. So bedbugs will only be killed within 3 to 7 days upon contact, and may take up to 2 weeks for complete eradication. So in the meantime, while waiting for this particular insecticide or this particular product to exert impact on the bedbugs population, the people in the house would continue to be bitten. If you look at the published information, it's actually quite promising. So this came from Kobe Shaw's lab in NC State. They evaluate Apprehend, that is the product that contained Beauveria bassiana, in comparison with Suspend SC that contained pyrethroids, deltamethrin. So as you can see, with the susceptible strain, you would find that the population would be controlled very quickly for the microbial insecticide as well as also the deltamethrin formulation. But when it comes to the pyrethroid resistant strain, you would find that the pyrethroid resistant strain would continue to survive just like the control, but with Beauveria bassiana, they will be able to crash the population at a quicker stage. Okay, so they conclude that the product containing Beauveria is effective against insecticide resistant bedbug. Botanical insecticides are also becoming increasingly popular. Generally, they contain essential oil, such as neem, cedar, clove, peppermint, etc. Now, in the US, many of these formulations against bedbug are exempted from EPA registration because they are considered under Section 25B. Okay, so they are not required to have an EPA registration number, and of course are exempted from EPA regulation on the tox as well as also efficacy. But at different states, they may require a state registration, okay, for this product. So if you look across the market or even online, you would find that there are tons and tons of such products. But I have to tell you that some of them are actually effective, especially as direct spray, but many others are actually not effective at all. Okay, so many of these products, they are effective as direct spray. So if you see the bug, you spray directly, you will kill the bug. And so would be many of the pyrethroid aerosol, okay, pyrethroid-based aerosol. But if you were to expect good residual effect with botanical insecticide, it is not easy, okay. Fumigants, such as Vicane, sulfuric fluoride, or methyl bromide. Methyl bromide has been phased out, but in some parts of the world, they are still being used. And also inert gases, such as CO2 and nitrogen, they have been used for bedbug control. So one of the nice things with fumigants is they are able to penetrate deep into many of these hidden area and harborages and kill all stages. The problem with fumigation is it's a major job. It requires a lot of plannings, and of course, there have to be a lot of safety measure involved when it comes to a fumigation job. For airplanes, bedbug fumigation, it's actually important, but that's the reason is because in the airplanes there are many regulations pertaining application of residue insecticides. So fumigation became a good choice, and still is a good choice when it comes to airplane bedbugs control in airplanes. The challenge with fumigation, major challenges, it's they do not have any residue activity. And of course, reinfestation could happen after the fumigation space is air. The penetration ability of inert gas is generally poor and normally would require longer time to be able to affect the population. So far, we have not found any resistance to fumigants in bedbugs. There have also been attempts to use ozone gas And this particular study was from the Purdue group. So Purdue Entomology has a professor that specialized on the use of ozone gas against stock product insects. So they thought that perhaps they should evaluate the use of ozone gas against bedbugs. But to kill 100% of adults and nymphs, very high concentration of ozone is required. So if you want to kill the eggs, even higher concentration is needed. So in the end, the authors conclude that the use of ozone to control bedbugs in homes is not feasible because of the logistics, the safety. Of course, ozone also could affect human at high concentration. And of course, when very high concentration is required, the cost factor is also an issue. Physical control, such as use of heat, cold, and physical removal of bedbugs has been attempted. And it's probably one of those approach that is effective, okay, in this current or present moment, especially heat treatment. It is effective, but it is also important to note that just like fumigation, so as soon as the room is air, you could have another bedbug infestation coming in, and then the infestation would continue. So there is no residue when it comes to physical treatment. The use of inorganic and mineral compounds, it's becoming popular, such as DE, dust, silica gel, and also limestone dust. This because they could absorb the wax layer of the bedbug's exoskeleton and causing the insect to dehydrate and die. Now, they will also remove the moisture from the bedbug harborage, so making them unsuitable for aggregation. So many of these products these days are available in dust formulation, but they are also aerosolized formulation. The action generally is slow, would take about 24 hours up to a few weeks to see 100% mortality of all stages. And generally, silica gel, it's more effective than DE dusts. But they are not suitable to be used in places where they have been constant air movement. There have also been efforts to combine the use of heat treatment with DE or silica dusts. And some people personally feel that if I had one, it's effective, but if I have two, that will be even better. Okay, so, but when you start using two, it is important to note that because when we use heat treatment, normally we need to use industrial fan to spread the heat throughout the entire room, especially when whole room treatment is being done. And this would kick up and also spread the dust around, and it could become an inhalation risk. Okay, so for such combination, if it's absolutely necessary, it is important that the PMPs would need to wear a proper gear, especially the respiratory mask during the job. What about the future? In academic research, there have been effort to look at RNAi, okay, where we silencing of the important genes, the genes that actually regulate molting, sperm release, for example, larvae development, reproduction or locomotion. So these are the RNAis that have been explored in different types of insects. In bedbugs, from Nebraska, they have looked at research on RNAi that will affect bedbugs reproduction, survivorship, and also insecticide tolerance. There have also been effort to look into coming out with a bedbug baits with the discovery of the blood constituent as phagostimulant by the NC State group. They have intensified the effort to develop a bedbug baits. So far, from the NC State group, there have been a number of papers they have been explored to look at the potential of a few toxicants that could lead to the development of a bedbug beds. But all these would remain a big challenge because we would still need a feasible and low-cost delivery system to apply this approach to the bedbug. And until then, all these remain as nice academic studies that are publishable, of course, good PhD research, but in terms of practicality, it remains to be seen. So one of the real challenge in managing bedbugs is actually the bedbug reservoir. So low-income housing is probably one of the worst, is probably having the worst bedbug infestation. So I'm sure Shang Lu Wang would be able to tell you many of these places, the tenants are so poor that they don't have the financial means to pay for any of this treatment. So they would be happy to accept whatever help, they could get from their management. And in most situations, pest control in these localities are paying very small or low dollar. And the PMPs may only be spending 2 minutes per house. So a lot of time, the control of bedbugs are not well carried out and they're not effective. And this basically causes bedbugs to propagate in massive number. And this actually serves as a bedbug reservoir that eventually would spread throughout the community. So for as long as bedbug reservoir remains there, we will not be able to completely get rid of the bedbug problem in the years to come. So lastly, insecticide resistance, especially pyrethroid resistance, is a major challenge to the management of both common and tropical bedbugs. So resistant management strategies such as the use of insecticide rotation and mixture could be used unless the population have metabolic resistance mechanism such as P450, which is very common, So biopesticide, desiccant dust, fumigants, physical methods, they are good options for managing bedbugs. Costs remain an issue for physical methods, especially when the fund allocation for pest management is so low, especially in low-income housing. So manufacturer will have to look at, we have to look into innovative formulation and delivery system. And lastly, the challenge of finding a low toxicity, cost-effective, and time-efficient management strategy remains. So I would like to acknowledge all members of the Urban Entomology Program at UCR that we work together as a team, and it's had been fun, working with everyone at the program in UCR. I would like to acknowledge my collaborator, Dr. Stephen Dodgett at Westmead Hospital in Sydney, my doctorate student, Leong Xingyan, I think she is in the Zoom today, and also Dr. Vera Singham, who is also my collaborator in University of Science Malaysia. So thank you so much for your kind attention. It has been an honor for me to be able to speak in this memorial seminar. Thank you, everyone.
Thank you, Chaiyan.
I have a question. If I, I'm going to jump in first because I know probably everyone has a lot of questions. I know you mentioned about RNAi and looking at the behavioral aspects of knocking out those genes. Do you know if anyone has done any gene drive experiments on kind of a sustainable way to knock out genes in populations?
I may not, from what I know, I have not seen any, but I may have missed out in the literature, but from what I have known, no.
Yeah, it makes sense that it probably hasn't been done. I know a lot of focus is on mosquitoes, and now they're trying to start gene drives in different systems. So I wondered if it had been done, but it probably has not.
That's right.
Thank you. That was I really enjoyed your talk.
Thank you. Thank you, Claude.
Chaiyan Lee, I've got a question.
Sure.
So when you showed the slide of fumigating the airplanes, I immediately triggered something. with brown marmorated stink bug that I've worked with a long time, New Zealand actually requires that if cars are imported to New Zealand from an area where the stink bug is present, they have to be fumigated or they can be heat treated. Now, it would probably be a monumental thing to do with an airplane, but I'm curious as to whether that's been thought about as an alternative to the fumigation.
You mean heat?
Yeah. My thought would be to roll the plane into a great big hangar that was set up to heat it.
Okay, yeah, this is excellent question. Now, a number of years ago, I had been engaged by a major airline in Asia, and they were having a lot of problems with bedbugs. So, and business class, passenger getting bitten and threatened to sue the airlines and all this mess going around. So what happened is they tried to go into, so we proposed the use of heat. And when we start talking to the engineers, it is not feasible because some of the epoxy, and the material used in the aircraft, especially in the cabin, they are heat sensitive. So anytime when you have a temperature that is above 55 degrees C, they start to change form. And that's why heat has never been explored for airplanes. And even the use of some of the insecticide, if you notice in airplanes, if you go to some places where they have disease vectors, they will be spraying those aerosol, but not applying it onto the seats and all that. So there are no residue spray on the seats. And the same reason is because of FAA regulation. Some of the material used in residue insecticide, the solvent, they could actually affect the material on the seats. And the last thing that they want, it's something that get detached, and during the decompression, that thing start flying and hit someone. Yeah. And that could be a big issue. So airplanes is a completely a different thing altogether when it comes to control of insects or even rats. So that's why fumigation is the preferred choice so far. It is a major task because many airlines, they prefer to have their planes on the air as much as possible because that will increase their profit. And the last thing they want to do is to have the airplane parked on the ground for 48 hours because of a fumigation exercise. Yeah.
Thank you.
We actually developed two programs, disinsection programs for two major carriers that have a hub center in the Northeast for that exact same reason, Dr. Lee. Yeah. And they call it disinsection programs that they develop, and it has to be approved by the FAA and all of that. That's right. And boy, they wanted it to work because right now all we do is fumigate. Yes. It's not cheap.
It's not cheap. Absolutely. It's an expensive exercise. You're right. Yeah. And not just that, there are not many countries that allow that to happen. So some airplanes has to be flown to certain countries to get the fumigation job done. And that actually is also expensive. Yeah. Especially when they required, so in the past, when they required to fumigate it with methyl bromide, because not all countries could, methyl bromide cannot be used in many countries these days. So they could only fly the airplanes to respective country where methyl bromide is still permissible.
Yeah, they applied for the QPS requirement for methyl bromide in the U.S. and it was denied. So it's only sulfurofluoride right now.
Exactly.
Quick question for you, Dr. Lee, on the work that you looked at regarding heat with the use of a amorphous silica gel. Is there any efficacy work that was done on that to see if the addition of a product like Cemexa would increase the results of heat? Because the problem that we seem to have with heat more than anything else is if the harbored spots are located centrally in the areas that are being heated, it's incredibly effective.
That's right.
Especially if monitoring is done well, you don't need anything else. However, if you end up in some of these infestations where it's either a bookcase up against the peripheral wall or in the wall void itself, those bugs have the time and the capability of moving away from the heat as the temperature is rising. So
That's
we've
right.
been using it that way. Does that make sense, or is there any data on that?
There have been some research work going on in UC Berkeley right now, and I know that they have been doing something, but so far the result has not been published. But this is an effective method, and it's an effective approach. But as I said, there are a lot of things involved when it comes to combination of this approach because of you need to take precaution when you use both heat and dust together. Yeah.
Dana, you have a question?
Yeah, thank you. So I saw you had in one of your later slides the work that Kobe Shaw was doing with the baits.
Yeah.
And from what I understand it, he was able to use those baits as a very effective delivery system for growth regulators, hydrometherpene and so on and so forth, that
That's
you
right.
can't spray. They very, very not efficacious when they're sprayed. Is your opinion that the baits are still a long way off in the future, or is this something that's going to be the future of bedbug control?
Excellent question, Dana. So the biggest challenge when it comes to bait is we need to attract bed bugs from a good distance, and that will require heat, that will require CO2. So if we put out a bait, we do not want just to put it out for 24 hours and that's it. We want something that could last at least for, let's say, a period of time, let's say 7 days. And to put out a product at a cost that is affordable, that could generate heat and CO2, and at the same time attract bed bugs from a distance, we have not seen any technology that could do that so far.
Okay.
Yeah.
So I'm curious what the bait actually looks like. Is this some kind of a liquid packet, or it's?
Yeah.
Yeah.
It is. Yeah.
Okay.
There have been attempts actually to design some form of small sachet, because bed bugs are attracted not really to the blood, but if you have ATP or even sodium chloride, they would feed on them already. But that particular pouch has to be warm, just like a human, and at the same time, they must have some form of attractant, kind of like CO2 from a distance that they could detect and move towards that particular bait. And that can be a challenge. They are not like roaches. Bedbugs are not like roaches because they would come out and feed. Roaches would go out and explore, unlike, bedbugs would probably spend time hiding most of the time. Yeah.
Thank you. That was a great talk.
Thank you.
Okay, one more question before we break. No more questions? Oh, the last chance. Okay, then thank you, Chaiyan,
Thank you.
for excellent seminar. So
Thank
for
you.
students who registered, we'll continue for another 1 hour. Okay, thanks.
Thank you.
Chaiyan, maybe we can have a 5 minutes break.
Sure.
You can come back around maybe 12 or 5.
Okay.
Okay.
5.
Right.
Sure. All right.
Mm.
Thank you.
So Chaiyan, normally at this time we would have lunch. So with students, who registered for the class.
That's right.
Yeah, so just a casual discussion. Whoever have questions with you and want to discuss.
Okay.
So we now have to be everything remote.
Yeah.
Yeah, it's just casual talk. We talk to just the students who is registered.
Yeah. Seminar thing over here also have changed a lot. In the past, normally